Good morning, everyone. Thank you so much for coming to the open day. My name is Veerle and this is Sara. We're both researchers here at the Department of Engineering Science. I also did my PhD in engineering at Oxford, so I've been around for a little while. What we both work on is leveraging the power of sound waves to tackle healthcare challenges, and that is very varied, from back pain to antimicrobial resistance, which we will tell you a little bit about later on. And this is actually, we made that yesterday with some of the bacteria that we work with. So what is ultrasound? Have you guys heard of ultrasound before? Yeah. So Lord, she's an artist, seems to think that that is an ultrasound picture. Do we agree? Do we think that that's ultrasound? No, exactly. That is very much not an ultrasound image. This instead is, which I'm sure many of you will have seen before, of a baby inside a mom's belly. Now, ultimately, this is just sound that's being sent into the body and reflected back, and we pick that up and we can create an image with that. And this sound is so high-pitched, such a high frequency that we can't actually hear it as humans. So how is that being generated? So you can think of it like a speaker with a moving front face, which basically can be mapped as this gray plunger that you see move. The result of that movement of the plunger is a condensing and a rarefaction of some of these particles that you can see. You see the denser regions and the less dense. Effectively, it's just a pressure wave. And that is all that sound is. That's even the sound that I'm speaking at you with. It's just a pressure wave. So positive pressure, negative pressure. And ultrasound is that, but very, very, very fast in frequency. Now, the cool thing about waves, or pressure waves in general, is that we can focus them. So very, very similar to using a magnifying glass to focus light, we can do the same thing using sound. I don't know, I am Dutch, so I did my schooling in the Netherlands, and we did that in primary school. But every time I mention it here, everyone is like, no, we don't do that in this curriculum. So if you haven't tried it, maybe don't try it at home. Anyway, we can do the exact same thing with sound. And one of the things that we use for that is this device over here. It is a ceramic ultrasound source or transducer, and it exhibits something called the piezoelectric effect. Have you guys heard of that before in physics? Yeah. So it's a very cool effect where an electrical stimulus is transformed into a mechanical one, and the other way around. So if we put an electrical signal on the ceramic, the response of that ceramic is to expand and contract. And very similar to that plunger I was showing you on the last slide, it creates this pressure wave. The focus of our sound source is similar to the light. It's about the size of a grain of rice. It's a little bit longer in the direction of the sound, and then much, much smaller, about 2 millimeters in the direction perpendicular to the sound propagation. And so this is one of the devices that we have in the lab. We often bring this to demos, but this is a bit easier on the slide. So I have on the left, in general, you can see the ultrasound source, like that one, sits at the bottom there, and it's pointing upwards towards the interface of the liquid and the air, just the room air. And if we send a small electrical signal, so that's the one on the left, we will start to see that, and you can see it a bit better from the bottom left, we start to see that the sound wave is pushing on that air-water interface. Now, if we increase the amplitude of our electrical signal, the result is that we start to get this vaporization. We start to see some steam. And you can see it even a bit better here, where you can see all of the steam. Now, do you guys think that this is hot? Any guesses? 50-50? Hot or not? Yeah, so it is actually cold. So there's two ways in which we can do this. And maybe you've seen this in chemistry in school already. But this is a phase diagram, in this case of water. Now, there's two ways in which we can go from a liquid to a gas. Either by changing the temperature. So if we increase the temperature, we get boiling, right? And it turns into a gas. Or we can drop the pressure. Now, and I told you before that ultrasound is a pressure wave. So positive pressure, negative pressure, positive pressure, negative pressure. And it's precisely that negative pressure that is dropping low enough that it actually pulls gas out of solution. Now, this is a phenomenon that is called cavitation. You will have all experienced that before, and Sara will tell you a little bit about that. But it's essentially the formation, the movement, and the collapse of gas bubbles.
So yeah, as Veerle mentioned, you probably have heard of cavitation before. So it was initially, I guess the effects of cavitation were initially discovered in ship turbines, where the rapid movement of ship turbines caused a negative pressure that creates bubbles. That actually caused a lot of problems when manufacturers were developing turbines, because it caused mechanical damage on the actual turbines. Also, boiling is an example of cavitation. In this case, this would be an increase in temperature causing bubbles to form. Ballistics can also result in cavitation, and that generally is the result of why gunshot wounds are so damaging. It's not the initial impact, but the cavitation that results from the negative pressure as a result of the rapid movement of the bullet. And then finally, this is probably what we're most familiar with. If you open up a can of really carbonated liquid, the rapid negative pressure that results in opening that can causes bubbles to form and explode, and can be very messy, as this young woman is experiencing. So one thing that we do in ultrasound is that instead of relying on the ultrasound wave to generate bubbles, we can actually just introduce bubbles into a system. And the way that bubbles respond to ultrasound works like this. So as Veerle mentioned, an ultrasound wave is just a positive and negative pressure wave. And in that environment, a bubble starts at a standard radius. In a positive pressure environment, it shrinks. In a negative pressure environment, it expands. And this oscillation occurs basically throughout the entire duration of the ultrasound wave. So this is an example of one of the bubbles that we would make in the lab under the exposure of an ultrasound pressure wave. And what's cool about ultrasound is that the bubble oscillates in a different way depending on how high the pressure is that we give it. So at a low-ish pressure, the bubble oscillates big and large forever. But if we increase the pressure, the bubble oscillation gets a lot more violent. And this violent cavitation activity, or bubble oscillation, can be used, in our case, for therapeutic purposes. And what we also do in the lab is also listen to the bubble activity in real time. So not only do we send sound to cause bubbles to oscillate, but that oscillation also generates sound. And we can listen to it and characterize it. And what this looks like in principle is, I don't know if you are familiar with looking at frequency spectra, but essentially you can think about it as all of these frequencies are kind of like tones, so like different keys on a piano. So at a low pressure, so that kind of oscillation, we get octaves of keys, which are known as harmonics. So if you're a music person, you're probably familiar with harmonics or octaves. But when we get that violent collapse, it's like hitting all of the keys at once. So it creates a lot of noise. And so we can listen to these bubble oscillations and characterize whether they're doing this kind of gentle oscillation or violent oscillation, and use that to kind of tune our ultrasound therapies. So it's useful to think about this also in terms of microbubbles are currently used as ultrasound contrast agents. So that kind of gentle oscillation, creating those harmonics or octaves, can be used to look at the microvasculature. In this case, this is a brain image of looking at ultrasound contrast agents flowing through microvascular networks in the brain. And then under high pressure, bubbles can both move and collapse. In this case, this is an experiment I did where I was looking at sending a really high-powered ultrasound pressure wave at a bunch of bubbles, which cause them to collapse and also to move. And this, you could imagine, could result in some really unique forces that we could harness for therapy. And so more on the therapeutic aspects that we are interested in. So bubbles can cause a number, well, I guess bubbles and ultrasound without bubbles can cause a number of effects that we can use in the body. So one is in which a bubble can oscillate next to a cell membrane, and that causes this process called sonoporation, where you can actually permeabilize cell membranes with bubbles. And this can look like this, where in gold is a cell membrane, a bubble's next to it, it causes that opening, and then it collapses eventually. And also here where the red is flowing into the cell membrane that was normally impermeable to that drug. And you can imagine that there are certain drugs that are difficult to pass through cell membranes, such as gene therapies, that could be really helped with this type of technology. Bubbles can also create really unique fluid forces around them, which can be used for basically pumping drugs beyond vascular spaces. It can also be used for heating. Veerle will talk a little bit about this in a couple slides, where you can actually use ultrasound to heat certain areas of the body based on thermal absorption of that kind of ultrasound energy. And then finally, if you really ramp up the pressure high enough, you can cause actual boiling at the focus of the ultrasound transducer, which is actually now being used clinically to ablate tumors. So that's a very exciting example.
Yeah, so we figured, now that we've given you a little bit of background in terms of what ultrasound can do other than just imaging, hopefully it's clear that a lot of engineering goes into these kinds of things that we do in the lab. But we figured we'll give you two concrete examples. So the first one will be a project that I have finished now, but it's on back pain. And then Sara will talk a little bit about antimicrobial resistance. So the project that I did, it is very close to my heart. That's a picture of my mother and my sister and me. My mother suffers from chronic back pain. This is actually an MRI scan of her spine. And what we can see is these are the bones, the vertebrae in the back, and in between are these black discs. They're the spinal discs. They're basically cushions that prevent the bones bashing together as you walk, which would be really painful. And in the case of my mom, especially the lower down you go, you see that they're very thin. So she actually doesn't have much of that shock absorbing cushion left, and it just causes her a lot of pain. Now, current treatments are limited, either physiotherapy, which is just symptom management, right, or very invasive surgery, which has questionable success rates. So we thought maybe we can engineer something that effectively can pump that cushion back up, and ideally do that without having to perform major surgery. So what we've come up with, it's a big collaboration. So we do the ultrasound part of all of this, but it's a collaboration with tissue engineering. We have come up with a material that consists of two components, mostly. One is a sugar solution, and the second one are really, really small lipid vesicles, a bit similar to the bubbles that Sara was talking about, but instead they're filled with a liquid. And these vesicles are filled with calcium, which we all know from milk, for example. Now, these vesicles have something really cool about them. They're engineered to be responsive to temperature changes. So as we heat these vesicles up to 41 degrees Celsius, so only just above body temperature, the lipids will change their conformation and actually start to leak the calcium out into the environment. That sounds bad, right? Leaky vesicles. But in this case, it's really good for us, because the calcium in turn is able to link up our sugars and actually form a network. So initially, this material is a liquid, which means we can just inject it through a needle. If we heat it up to 41 degrees Celsius, it will start to leak calcium, cross-link or link up our sugars and form a network, or in other words, a gel. Now, and that heating is something we can do with ultrasound, and that can be completely non-invasively from outside the body, similar to that magnifying glass with that high energy spot that I was showing earlier. Now, in order to do that, we had to design our experiments. So we use a lot of different equipment, and you can see me preparing it here. This is where the ultrasound source sits. There is a whole bunch of amplifiers, function generators. Maybe you've seen some of that in some of your physics classes that are attached to this. And then I was recording the temperature, because obviously I wanted to know what was going on. And this is what that then looks like. So we've got time on the x-axis, temperature on the y-axis. I wrote an algorithm, so again, very much engineering that we do, to control my ultrasounds, my treatment, based on the temperature feedback that I was getting. So every time I was hitting what I had put as my upper limit, it would turn it off, the temperature would drop, turn it back on, etc., until 5 minutes till completion. And this is one of the gels that we got out of that. So that looks great. We were very happy with it, a bit like a sweet. And then they are tested mechanically, because obviously we want to make sure that they are good enough to be able to replace spinal disc tissue in a human. They're not quite strong enough yet, so we're on our way there. And one of the cool things that we observed in this experiment particularly is that we could actually hear this gelation happening. So Sara was telling you that these bubbles that are being generated, they also produce a sound. And the piezoelectric effect, as we talked about before, goes both ways. So the pressure wave that these bubbles generate can be picked up by a ceramic, turned into an electrical signal, and that's how we measure the sound. Now, normally this is, again, such high frequencies that we can't hear it. I transformed it for this presentation so you can actually hear it. We're going to see, I think it will play the sound from my laptop, so we're going to find out if that works. Perfect. I'm going to play you that one more time, but this is sound from a sample that doesn't contain the calcium. So it will always remain a liquid. And as Sara was saying, it's like hitting all of those keys at the same time, so it is just noise. So I'm going to play that for you one more time. Now instead, I'm going to play you one more, which is a sample that does actually contain the calcium, meaning that when we heat it up, it will start to leak out, cross-link our sugars, and it will form a gel. Now, I want you guys to tell me if you can hear the difference. What are we hearing? It goes down, exactly. So it literally goes quiet. And the way to think about that is these bubbles are moving in a liquid and they're absolutely fine. They can move quite freely. But as that liquid is gelling and becoming stiffer, that movement of these bubbles are reduced. So they just can't move as much and therefore don't produce as much sound. And then the last step in that project is just to check whether we can do this in a more clinically relevant model. So we did the exact same experiment, but in this case, we injected our material into cow spines from the butchers. I'm a vegetarian, so it's not my favorite part of my project. But we did actually see partial restoration of our biomechanical function in that spinal segment for all of our samples tested.
You may be familiar with antimicrobial resistance, which is a massive global health problem that has emerged in the last couple decades, where essentially antibiotics that used to work on infections are no longer effective. And this can have many different reasons. So there's genetic changes that bacteria can undergo that make antibiotics less effective. Bacteria evolve on such a much faster rate than human evolution that they're able to rapidly gain genetic advantages that make them less susceptible to antibiotics. But kind of on a macro scale level, bacteria are also really good at forming communities that can protect them from their environment. And this can be both physical protection, so literal creating substances that form a barrier for antibiotics to get through, or also functional protection, where the bacteria can be somewhat dormant, which means that certain drugs may not work on them, or it means that they can rapidly exchange genetic material to make resistance rates go up really rapidly within these environments. You may be, and so we call those communities of bacteria or microbes that exist within this environment a biofilm. And you're probably already familiar with biofilms. Dental plaques are an example of biofilms that your dentist is probably trying to get you to floss and brush your teeth to not get. I would really recommend doing that because they're really gross and can cause lots of problems. But so these are kind of the types of treatments that we're looking at affecting. So I've been looking at a couple different ways at using ultrasound and microbubbles to break up biofilms to get more effective antimicrobial therapies. So the system that I look at is similar to Veerle's, where I have a therapeutic ultrasound source, so that similar kind of curved piezoelectric element that you could see creating the steam. I use that and focus it towards a biofilm, which I grow in small flow channels. I add microbubbles to potentiate that effect of ultrasound. And then I can look at a couple different bacterial species. So the ones that I've focused mainly on are a species called Staphylococcus aureus. So you might be familiar with staph infections, or MRSA is really common. And it's a really nasty pathogen that has gotten a lot of resistance recently in the past few decades. And then also Pseudomonas aeruginosa, which is another pathogen that is super problematic, and they form kind of different types of biofilms, so it's an interesting comparison to look at these two. And then I can put them in these flow channels, and this is an example. Also, in this case, these green kind of things are bacteria. I just stained them green, so that's just all of the bacteria existing on the surface. And in these two images, that's a scanning electron microscopy image of the bacteria, so you can see they're quite clearly in these very dense communities. And then I can also stain them pink. In this case, we're just going to look at how it looks like under the, with just a camera, how it looks like when ultrasound is exposing these biofilms. So you can see there's kind of an area in the center that clears. Is there like a little clicker on here? No. Is there?
Yeah, there's that one.
That little guy. Okay. Yes. So this area in the center goes from kind of pinkish to a little bit more whitish. And it's kind of an interesting pattern that forms there. And the reason that happens is that the actual pressure within that channel looks like this, where we have areas of high pressure and then areas of kind of lower pressure. So Veerle showed earlier that an ultrasound pressure kind of distribution looks kind of like a grain of rice. And you can imagine that this grain of rice is existing within this channel. It's just getting also kind of distorted by waves reflecting off the top surface and down and top and down, and it creates kind of some weird patterns. What's cool is that the kind of overall distribution of biofilm removal looks quite similar to the pressure distribution within the channel. So that's good on me for aligning it well. And we can also look at kind of more realistic substrates as well. So some of the work that I've been doing more recently is growing infections on porcine skin. So similar to Veerle, I call up the local butcher and ask to have some skin. Very gross, but it forms a nice representative biofilm. And then we can also look at this under the exposure of ultrasound. So this is the skin, ultrasound's coming this way, and then you can see that biofilm really nicely coming off of the skin piece, meaning that this could be a useful technique in eliminating infections. And then we can also see it just on a graph where we can look at the number of live bacteria in each of the skin pieces, and after ultrasound it goes down, which is exciting. So we've talked today how ultrasound is more than just babies. We can use it for targeted pain management using the back pain project. We can use it to treat antimicrobial resistance, but then there's tons of applications that we haven't talked about. So ultrasound can be used to stimulate bone growth or other types of tissue engineering applications. It's used for cancer drug delivery or cancer tumor ablation. Other groups or other lab members in our group are working on needle-free vaccination with ultrasound to eliminate the pain associated with getting a needle vaccine. It can be used for sonothrombolysis, which is basically breaking up blood clots within arteries. And also, one of the kind of newest and some of the most exciting applications is actually in the brain, where you can push drugs beyond the blood-brain barrier to make drugs that previously weren't accessible to the brain now accessible. And also neuromodulation is another kind of exciting application where the ultrasound pressure waves can actually stimulate neurons to do a variety of different types of neuromodulation applications.
Yeah, so with that, we just wanted to say thank you so much for listening to us. We actually run an Instagram channel. We always plug it because it gives you a little bit of insight into what a biomedical engineer does day to day. We even got our boss here. She's one of the professors here, so if you do come and study engineering here, you'll see her in a more professional capacity too.
Tell her that you saw her on Instagram first.
She'll love that. So if you do want to give us a follow, feel free, or reach out to us. We get a lot of students coming through the labs, etc. We don't have any places anymore this year, but if it's something that you're interested in, feel free to reach out to us. And with that, we just want to say thank you, and we're super happy to take any questions.